Okay, today we're going to go over the basic anatomy of the muscle fiber and the action potentials. Tomorrow and Monday, you're going to be doing a lab on how muscles are organized and how they are formed. And then we will start building our muscles on your little mannequin. Okay. So let's go. The functions of the muscular system is body movement, maintenance of posture, keep you upright, respiration. What muscle allows you to respirate or breathe? Is your lung a muscle? A diaphragm. It's your diaphragm that contracts and changes the pressure. It decreases the pressure inside the lungs. So the high pressure outside your body goes into your lungs, and when the diaphragm then relaxes, it pushes the air back out of your lungs. The production of body heat, whenever your muscles move, it produces heat. So when you get really cold, what's one of the first things you start doing? Shivering, which is small muscle contractions that would produce body heat. Communication, it can send electrical signals down a muscle fiber. Constriction of organs and vessels, can you think of an example of that? Smooth muscle in your stomach, intestines, that moves food through, yep. And then your heartbeat. Awesome. So some general functional characteristics is it's able to contract, contractility. So it's the ability of muscle to shorten with a force. That's how you get any movement of skeletal muscle. Excitability is the capacity of a muscle to respond to a stimulus. So a nerve has to come and stimulate that muscle and have an electrical potential go down that. Extensibility is a muscle is able to be stretched to its normal resting length and beyond to a limited degree. So it contracts in order to go backwards. It extends out, and it can go past a little bit its resting stage. And then elasticity is the ability of a muscle to recoil to its original resting length after it's stretched. So when you do stretching before you do any exercising, you're kind of stretching it past its resting length, and then it goes back into it. That's why you don't want to overstretch a lot. You can hurt your muscle, tear muscle. There's 3 different types of muscle tissue: skeletal, smooth, and cardiac. Skeletal muscle is responsible for locomotion or movement, facial expressions, posture, respiratory movements, and other types of body movements. So we are going to learn the first muscles we're going to build are your facial muscles. We're going to learn the different muscles that give you facial expressions, like lifting your eyebrows or smiling or bringing your nose up. And we're actually going to make an eye movie where you're going to make all those facial expressions. Okay. And then you also have some voluntary, skeletal muscles are always voluntary. And voluntary means you have control over them, right? You can tell your arm to move. Your arm doesn't just jerk out of involuntary control. Sometimes with your siblings it does. Smooth muscle is in the walls of hollow organs, the blood vessels, the eye. Where is there muscle in your eye? Not the retina. Well, when you move your eye left to right and up and down, that's all skeletal muscle. You have control over that, right? Smooth muscle is involuntary. No, because you have control over that. That's all skeletal muscle. Ah, not your pupils. Your pupils are the whole. It's your iris. Your iris is actually a muscle, and it contracts, dilates, and constricts. There. Oh. Whoops. Does this look different than it just did? No? Okay. Smooth muscle in the walls, yes, the eyes, the glands, in your skin. What are the muscles in your skin that are smooth? Erector pili that we've looked at, that gives you goosebumps. And some functions is to propel urine, mix food in the digestive tract, dilating and constricting the pupils, and restricting or regulating blood flow. In some locations, it's autorhythmic. That would be like in your intestines, how it's called peristalsis, but it's wave-like movement, and it just is always kind of in a rhythmic motion, moving the food through. It's controlled involuntary by the endocrine and autonomic nervous systems. Then cardiac is just in the heart, and it's the major source of movement of blood. It's also autorhythmic. You have a regular heartbeat, a rhythmic heartbeat, and it's controlled involuntary also. So these are the different skeletal, smooth, cardiac, kind of compares all of them. That's from your book. So skeletal muscle structure. It's composed of muscle cells. We've kind of looked at in the tissue chapter of the muscles, right? It's connective tissue. It has blood vessels and nerves within it. It needs a lot of oxygen from your blood, and the nerves is what actually can make the muscle contract. Fibers are very long, cylindrical, and multinucleated, meaning it has several nuclei within the one cell. They tend to be small in diameter in small muscles and larger in large muscles. They develop from myoblasts, and the numbers remain constant. You just increase the size of them. They're striated in appearance due to light and dark banding. And we will look at the bands on the muscle, but that you can see the dark bands running through that. That's the striations. Okay, the muscle is all covered with some connective tissue, and there's different layers. The epimysium is connective tissue that surrounds the whole muscle. The perimysium surrounds a group of muscle fibers, and then the endomysium surrounds each muscle fiber itself. So if we look at this picture, the epimysium would cover the whole outside of this whole muscle here. The perimysium would just surround this muscle bundle, a group of muscle fibers, and then endomysium covers just that one fiber. Like that one right there, there'd be endomysium around that fiber, The fascia is a connective tissue sheet. It forms a layer under the skin. It holds the muscles together and separates them into functional groups. So like it separates your deltoid from your biceps and your triceps. Okay. Have any of you guys heard of fasciitis? A lot of times you'll hear of plantar fasciitis, like on the bottom of your foot. Gets an inflammation there. Allows free movements of muscles. They're able to kind of slide past each other. And it carries the nerves and blood vessels and lymphatic systems there. And it continuous connective tissue with the tendons and periosteum. So it's kind of attached to the periosteum, and then it goes into the tendon, and then it connects to the muscle. So it actually is all connected. It's not like attached. It's just one continuous string of fibers. Goes from your where is your periosteum? What is periosteum? Yep, it's the outside layer of your bone. So the tendon connects your muscle to bone, so it's a connective tissue that goes from the outside of your bone into the tendon and then into your muscle. The nerve and blood vessel supply. The motor neurons stimulate muscle fibers to contract. And then the nerve cells with cell bodies or brain in the brain or spinal cord, they have axons, extend to the skeletal muscle fibers through nerves. So you just have long axons reaching to the muscle. The axons can branch so that each muscle fiber is innervated. And then we also have capillary beds. So here in this picture, we can see the capillary beds is the blue and red, and then this yellow is the nerve fiber, or the nerve. And here it shows the branching so that each of these fibers have its own innervation there by the axon. And the muscle fibers, the nuclei is just inside the sarcolemma, which is just the outer membrane of the muscle. The cell is packed with myofibrils within the cytoplasm. Cytoplasm is just the fluid inside that muscle fiber, that muscle cell. The myofibrils are thread-like, and they're composed of protein threads called myofilaments. There's actin and myosin, and we're going to look at how they work together. The sarcomeres are highly ordered repeating units of myofilaments. So you have these myofilaments, and they're in bands, and they extend. We'll have one myofilament here, and then it's just a repeating, and the repeating bands of the myofilaments is what make up the sarcomere. So we'll look at a picture, I think, on the next slide. Can I switch it? Second. Yes, here we go. So here we have our muscle fiber, and these are the myofilaments. This is the actin myofilament, the purple here. The green is the myosin. So this right here is the myofilament right here, and then it's repeating so that this is the sarcolemma as it repeats down right there. Actually, it'd be right there because that's the muscle fiber. So these actin and myosin filaments act together to make a muscle contraction. And if the purple is actin, what do you think the actin kind of looks like? What? A little bit. Grapes? What they a lot of times they're referred to as like a string of two strings of pearls. So if you take a pearl necklace and you have it stretched together and then you twist it upon itself. That's kind of what actin is like, looks like. And then what does the myosin look like? Pac-Man? It's the green. Ah, a golf. It looks like golf clubs, right? Kind of like golf clubs, two golf clubs, and then is that, what's the long part of the golf club called? Is the shaft? I'm not a golfer either. The head? Isn't that what you hit the golf ball with? Yes? Okay. So they're like twisted. You have two golf clubs, and then you twist them along each other. That's kind of what the myosin looks like. Okay. And then we actually have, it's called tropomyosin and troponin. We'll look at those, the blue that just kind of circles within the groove of the actin, and then the red, and we'll see what those do also. This is troponin and tropomyosin. This is a really good figure to know. I don't know what figure it is in your book, but it shows everything really good. So we have two strands of fibrous actin that form a double helix, like DNA. Extends the length of the myofilament and is attached at either end of the sarcomere. It's composed of G-actin monomers, each of which has an active site. So each one of those beads is called a G-actin monomer. And then it's just repeating itself. Each of those little round balls have an active site. And what happens is that active site of the actin can bind myosin during a muscle contraction. Okay, and then I said the tropomyosin is this blue cord. It kind of runs around the groove. It's an elongated protein that winds around the groove of the actin double helix. And what happens is when this, well, let's talk about the troponin first. The troponin is this red part, and you can see it's repeating along the actin filament also. It's composed of 3 subunits. One binds to actin, one binds to the tropomyosin, and then one binds to calcium ions. And what happens is when calcium binds to the troponin, that changes the shape of it. It allows the troponin to move the tropomyosin, and the tropomyosin then exposes the active site on the actin. So, for a muscle contraction, calcium binds to troponin. That moves the tropomyosin to expose active sites on the actin, which then the active sites bind the myosin. Okay? I'll show you a little, we're going to talk about the myosin next, and then I'll show you a little demo of that. So in a myosin, they're called the thick myofilaments. Actin's thin, myosin is called the thick myofilament. There's many elongated myosin molecules that are shaped like golf clubs. So the molecule consists of 2 heavy myosin molecules wound together to form a rod portion, and then 2 heads. And those myosin heads bind to active sites in the actin, and they're called cross bridges once they bind. They attach the rod portion by a hinge region that can bend and straighten. So it's like if you had your golf club, and the golf club the head could come up and down. It was on a hinge. And then it has ATPase activity, so it can break down ATP. And part of that energy is used to bend the hinge region of the myosin during a contraction. So ATP needs to bind there. When ATP is broken, it releases energy and allows that golf club head, the head of the myosin, to change shape. And so let's So the kind of demo that I always learn is if this actin, it's like a round ball but has an active site. When the active site is exposed, it's kind of like it has a spot right where myosin can fit in. So if myosin can fit in there, and then when the ATP is broken and it gives off energy, the head cocks back and moves the actin. And then when the ATP releases, it releases off the active site and it moves again. And the head goes back, it cocks back. So this is how a muscle contraction works. This is my actin, this is the myosin. Connects, it moves, it releases, and then it goes. All that happens really fast when you have a muscle contraction. Okay? Didn't know all that happened in your muscle, did you? So the sarcomere is from Z disc to Z disc. And what these Z discs, I think there's a bigger picture next, but these blue zigzag lines, those blue zigzag lines they serve as attachment for actin filaments. So all these actin are bound to the Z disc. Here's a Z disc, here's a Z disc, here's a Z disc, okay? They're striated because of these different bands. So the I band extends from the Z disc to the end of the thick filament. The thick filament is which protein? Which myofilament? The thick is the myosin. So if it extends from the Z disc to the ends of the thick filament, this would be the Z disc, extends from the Z disc to the end of the thick filament. This is going to be a better picture. You can't read those very well. The A is the length of the thick filament. So from wherever the myosin is to this myosin head, here's the A band. The H zone is the region in the A band where the actin and myosin do not overlap. So right here where there's no overlapping, that's called the H zone. And then the M line is the middle of the H zone. Right here, right down the middle of the myosin. We're going to look at a bigger picture of this so you can see it a little better. So the Z disc is right here. This blue line right here. The I band is from wherever the actin is without any myosin. I band. A band is from myosin, from the end of one myosin to the end of that same myosin here. The H zone is where there's no overlapping of any of the, in the middle of the actin along the myosin where there's no overlapping of actin and myosin. And the M line is right down the center of the H zone. You guys are going to need to know those zones. This sliding filament model is what I was showing you with the myosin and actin moving back and forth. The actin myofilaments slide over the myosin to shorten the sarcomeres. So as a contraction occurs, that myosin is moving along those actin molecules and the Z disc get closer and closer together. So the actin and myosin don't change length, actually. The shortening of the sarcomere is responsible for a skeletal muscle contraction. And then during relaxation, the sarcomeres lengthen because of some external force, like a contraction of an antagonistic muscle. So when you bend your arm, you're contracting this muscle. Can you just relax it without contracting another muscle? No. When you move it down, what are you contracting? Your tricep then. So you can't actually just relax a muscle by itself after it's been contracted. You're actually going to use an antagonistic muscle to lengthen that muscle again. That's what that means. So here we can see that as the Z bands get closer and closer together, the H zone gets smaller and smaller, that yellow portion, until it's closed. The A band, the pink part, gets longer and longer as it shortens. There. And I think that's where we'll stop. Okay. That's a lot of info, isn't it? Okay. So you guys have any questions on muscle fibers, the actin myosin, the sliding filament model as of yet?
Not right now.
Not right now. Okay.